So here's something worth thinking about as you settle in tonight. Right now, inside your body, somewhere around 300 billion cells are in the process of dividing. 300 billion. That number is almost impossible to hold in your mind. But let's try anyway. If you counted one cell division per second, it would take you about 10,000 years to count them all. And that's just what's happening today. Tomorrow, another 300 billion. The day after, another 300 billion. Your body is a construction site that never shuts down, never takes a holiday, never even pauses for a coffee break. And the vast, vast majority of the time, this works flawlessly. The copies are clean. The new cells do exactly what they're supposed to do. They show up, they do their job, and eventually, when the time comes, they die on schedule and make room for the next generation. But every once in a while, and this is really the whole story we're going to explore tonight, something goes wrong with one of those copies. A tiny error slips through. A letter gets changed in the instruction manual. And most of the time, even that doesn't matter, because your body has layers upon layers of backup systems designed to catch mistakes and fix them, or if they can't be fixed, to quietly destroy the damaged cell before it can cause any trouble. It's an unbelievably elegant system. But cancer is what happens when all of those safety nets fail, one after another, in the same cell. It's not one thing going wrong. It's a whole series of things going wrong, in just the right combination, in just the right order. And understanding how that happens, really understanding it from the ground up, is one of the most fascinating stories in all of science. So let's start at the very beginning. And I mean the very beginning, not with modern oncology or chemotherapy or genetic sequencing, but with the first time a human being looked at a tumor and tried to make sense of what it was. The oldest known description of cancer comes from ancient Egypt, from a document we now call the Edwin Smith Papyrus. It dates to around 1600 BCE, but scholars believe it's actually a copy of a much older text, possibly written around 3000 BCE by Imhotep, who was the chief physician to the pharaoh Djoser. Now, Imhotep is a fascinating figure in his own right. He was an architect, an engineer, a priest, a physician, and he's sometimes considered the first doctor whose name we actually know. The papyrus that bears the name of Edwin Smith, the American antiquarian who bought it in Luxor in 1862, describes 48 surgical cases in remarkable clinical detail. Broken bones, dislocated jaws, wounds to the head. It's methodical and practical, almost modern in its approach. But case number 45 is different. It describes bulging masses in the breast, tumors, and the unknown author's assessment is blunt and haunting. Under the section where treatment would normally go, it simply says, "There is none." Think about that for a second. Nearly 5,000 years ago, someone looked at cancer and recognized it as something fundamentally different from other diseases. Not a wound that could be stitched, not a bone that could be set. Something else entirely. Something that defied the tools they had. The word cancer itself comes much later, from the Greek physician Hippocrates, around 400 BCE. He used the word karkinos, which means crab. And there's some debate about exactly why he chose that image. Some historians think it was because the swollen veins radiating outward from a tumor looked like the legs of a crab. Others think it was because of the way the disease grips the body and won't let go. Either way, the metaphor stuck. The Roman physician Celsus later translated it into the Latin word cancer, and that's the name we still use today, 2,500 years later. But here's the thing. For most of human history, nobody had any real idea what cancer actually was. They could see it, they could describe it, they could sometimes cut it out, but they couldn't explain it. The dominant theory for over 1,000 years was the humoral theory, which came from Hippocrates and was later expanded by Galen, the enormously influential Greek physician who practiced in Rome around 150 CE. Galen believed that the body contained 4 humors: blood, phlegm, yellow bile, and black bile, and that disease was caused by an imbalance among them. Cancer, in Galen's view, was caused by an excess of black bile. Too much of this dark, melancholic fluid would accumulate in one spot and form a tumor. Now, this was completely wrong, of course. Black bile doesn't even exist as a distinct substance. But Galen's authority was so immense, his writing so comprehensive, that this theory dominated medical thinking for well over 1,000 years. And it had real consequences. Because if you believed cancer was caused by a systemic imbalance of fluids throughout the entire body, then surgery didn't make much sense. You could cut out the local tumor, sure, but the underlying imbalance would still be there, and the cancer would just come back somewhere else. So for century after century, the main treatments for cancer were dietary changes, bloodletting, and various purgatives designed to rebalance the humors, none of which worked, obviously. But the theory was so deeply entrenched that it took an extraordinary amount of evidence and courage to challenge it. The first real cracks in the humoral theory didn't appear until the Renaissance, when anatomists finally started opening up bodies and looking at what was actually inside. Andreas Vesalius, working in Padua in the 1530s and 1540s, performed meticulous dissections and found no evidence whatsoever of black bile. He looked for it. It wasn't there. His monumental work, De Humani Corporis Fabrica, published in 1543, was one of the great turning points in the history of medicine, because it established that you had to look at the actual body to understand disease, not just read ancient texts. But even Vesalius didn't have a better explanation for cancer. He just knew the old one was wrong. And that's a really important moment in any field of knowledge, by the way. The moment when you know the old answer is wrong, but you don't yet have a new one. It's uncomfortable. It's destabilizing. But it's where real progress starts. The next big leap came from a place you might not expect. In 1761, a London physician named John Hill published a short pamphlet noting that people who used excessive amounts of snuff, powdered tobacco that you inhale through the nose, seemed to develop cancers of the nasal passages at unusually high rates. This was, as far as we know, the first time anyone had linked cancer to a specific external substance. It was a small observation, almost a footnote, but the idea behind it was revolutionary. Maybe cancer wasn't caused by some mysterious internal imbalance. Maybe something from the outside was causing it. Then, in 1775, a surgeon named Percival Pott made an observation that would change the entire trajectory of cancer research. Pott worked at St. Bartholomew's Hospital in London, and he noticed something peculiar about chimney sweeps. These were often boys, sometimes as young as 4 or 5, who were sent up into chimneys to scrape out the soot. It was brutal, dangerous work. And Pott noticed that an alarming number of these boys, as they grew into young men, developed cancer of the scrotum. He called it the chimney sweep's cancer, and he argued that it was caused by prolonged exposure to soot, specifically by soot lodging in the folds of the skin. Here's what's remarkable about Pott's observation. He didn't know about carcinogens. He didn't know about DNA. He didn't know about mutations. He was working more than a century before anyone would understand the mechanism by which soot causes cancer. But he got the essential insight exactly right. Something in the environment was entering the body and causing normal tissue to become cancerous. That's an incredibly modern idea for 1775. And it had immediate practical implications. In Denmark, where chimney sweeps began bathing regularly and wearing protective clothing, the rates of scrotal cancer dropped dramatically. In England, where working conditions were slower to change, the disease remained common for decades. So by the late 1700s, there were these two threads starting to emerge. One was the anatomical thread, the idea that cancer was a physical thing in the body, a growth, a mass of tissue that could be studied and described. The other was the environmental thread, the idea that something from the outside could trigger cancer. But what was still completely missing was any understanding of what cancer actually was at the cellular level. What was this tissue made of? Why did it grow? Why didn't it stop? That question couldn't even begin to be answered until someone invented the tools to look at cells. And that brings us to the microscope and to a German pathologist named Rudolf Virchow, who in the mid-1800s would fundamentally reshape how we think about disease itself. Virchow was born in 1821 in a small town in what is now Poland, and he was, by any measure, one of the most important figures in the history of medicine. He was brilliant, combative, politically radical. He once got into a duel with Otto von Bismarck, which is a story for another time, and he was absolutely relentless in his insistence that disease had to be understood at the level of the cell. His great insight, published in 1858 in a work called Cellular Pathology, was captured in a Latin phrase, omnis cellula e cellula. Every cell comes from a cell. That sounds almost obvious now, but at the time, it was a thunderclap. It meant that disease wasn't caused by bad air or imbalanced humors or supernatural forces. Disease happened when cells went wrong. And cancer, Virchow argued, was a disease of cells, cells that were dividing when they shouldn't be, growing when they should have stopped. Now, Virchow got some things wrong. He believed that cancer was caused by chronic irritation, that if you irritated tissue enough, the cells would eventually start growing out of control. This wasn't entirely wrong, actually. Chronic inflammation really does increase cancer risk, and we understand the mechanisms for that now. But it was far from the complete picture. Still, Virchow's fundamental framing, that cancer is a disease of cellular behavior, was exactly right, and it's the framework we still use today. And here's where it starts to get really interesting, because once you establish that cancer is about cells behaving abnormally, the next question is obvious. Why? What makes a normal cell, a cell that's been following the rules its entire life, suddenly start breaking them? What changes inside the cell to flip that switch? The answer to that question would take another 100 years to fully emerge, and it would require breakthroughs in genetics, molecular biology, virology, and biochemistry. It's a story that involves radiation, mustard gas, viruses that cause tumors in chickens, a pair of scientists who figured out the structure of DNA in a pub in Cambridge, and a quiet researcher in the 1970s who discovered that the genes capable of causing cancer aren't foreign invaders at all. They're already inside every single cell in your body, sitting there, waiting. They're part of you. They always have been. But to get to that story, we need to talk about what was happening in the early 1900s when a young pathologist named Peyton Rouse was working at the Rockefeller Institute in New York, and a farmer brought him a chicken with a tumor that had a strange, firm mass growing on its breast. This was 1910, and Rouse was only 30 years old, still early in his career, still trying to make a name for himself. The chicken was a Plymouth Rock hen, and the tumor was a sarcoma, a cancer of the connective tissue. Now, most researchers at the time would have examined the tumor, maybe written up a description, and moved on. But Rouse did something different. He took a piece of that tumor, ground it up, passed it through a filter so fine that no cells, not even bacteria, could squeeze through, and then injected the filtered liquid into a healthy chicken. And the healthy chicken developed the same cancer. Think about what that means for a second. He had removed every cell from the extract. There was nothing alive in that liquid, at least not by any definition of life that scientists used at the time. And yet whatever was in that filtered fluid could cause cancer in a perfectly healthy animal, something invisible, something smaller than a cell, something smaller than a bacterium, was capable of triggering a tumor. What Rouse had isolated, though he didn't fully understand it yet, was a virus. It would eventually be named the Rouse sarcoma virus, and it would become one of the most important discoveries in the entire history of cancer research. But here's the thing. Almost nobody believed him. When Rouse published his findings, the scientific community largely shrugged. Some were skeptical, others were outright dismissive. The idea that a virus could cause cancer seemed bizarre, almost absurd. Cancer was supposed to be a disease of cellular misbehavior, a problem of growth gone wrong. Viruses caused infections, colds, flu, things like that. The two categories didn't seem to belong together. And to make matters worse, Rouse could only demonstrate this effect in chickens. He couldn't show the same thing happening in mammals, let alone humans. So the work was seen as a curiosity, an oddity specific to poultry, not something with broad relevance to understanding cancer in people. Rouse was so discouraged by the reception that he actually stopped working on tumor viruses for years. He moved on to other research. Decades passed, and it wasn't until much, much later, 1966 to be exact, that the Nobel Committee finally awarded Peyton Rouse the Nobel Prize in Physiology or Medicine for his discovery. He was 87 years old. 55 years had passed since the original experiment with that Plymouth Rock hen. It's one of the longest gaps between a discovery and its Nobel recognition in the history of the prize. And by the time Rouse received that award, the scientific world had changed enormously, because researchers had finally begun to understand why his discovery mattered so much. Not because viruses cause most cancers. They don't, actually. Only a fraction of human cancers are directly caused by viruses, but because of what the Rouse sarcoma virus would eventually reveal about the genetic machinery of cancer itself. That revelation, though, was still decades away. So let's pause on the timeline for a moment. We're in the early 20th century. Virchow has established that cancer is a disease of cells. Rouse has shown that at least some cancers can be triggered by a virus, but nobody really understands the mechanism. Nobody knows what's actually happening inside the cell to make it cancerous. And meanwhile, something else is happening in the world that's about to become terribly relevant. People are discovering radiation. Wilhelm Röntgen discovered X-rays in 1895, and within months, people were using them for everything. Looking at bones, fitting shoes, entertaining party guests. It sounds reckless now, and it was. Nobody understood the danger. Marie Curie was carrying vials of radium in her pockets, and her notebooks are still so radioactive today that you have to wear protective equipment to handle them. Early radiologists developed horrible burns on their hands. Some of them developed cancers. By the early 1900s, it was becoming clear that radiation could cause cancer, though the mechanism was completely unknown. This was an important clue, even if nobody could interpret it yet. Something about radiation was changing cells in a way that made them grow uncontrollably. The same was true of certain chemicals. Remember Percival Pott and the chimney sweeps, or John Hill and the snuff users. Soot, tobacco, radiation. All of these external agents seemed capable of triggering cancer. But how? The answer would require understanding something that scientists in the early 1900s were only just beginning to glimpse: the gene. Now, the word gene was coined in 1909 by the Danish botanist Wilhelm Johansen. But the concept was older than that, going back to Gregor Mendel's pea experiments in the 1860s. By the early 20th century, scientists knew that hereditary information was carried on chromosomes, those thread-like structures inside the cell nucleus. Thomas Hunt Morgan, working with fruit flies at Columbia University, just across town from where Rouse was working with his chickens actually, was mapping genes to specific locations on chromosomes. The idea was taking shape that genes were discrete units of information that controlled specific traits. But what genes were made of, physically, at the molecular level, that was still a mystery. And here's where the story takes one of those beautiful, winding paths through seemingly unrelated fields before everything converges. Because to understand cancer, you ultimately need to understand DNA. And to understand DNA, you need to follow a completely different thread of scientific history, one that involves a British medical officer, a quiet biochemist, and eventually that famous pair in a Cambridge pub. In 1928, a British bacteriologist named Frederick Griffith was studying pneumonia. He was working with two strains of Streptococcus pneumonia, one that was virulent, meaning it could kill mice, and one that was harmless. The virulent strain had a smooth polysaccharide coat that protected it from the immune system. The harmless strain lacked this coat and looked rough under a microscope. So scientists called them the S strain and the R strain, smooth and rough. Griffith did something clever. He killed the virulent S strain with heat, so it was completely dead, and then he mixed the dead S bacteria with the living but harmless R bacteria and injected the mixture into mice. Now, logically, nothing bad should have happened. The dangerous bacteria were dead. The living bacteria were harmless. But the mice died. And when Griffith examined their blood, he found living S-type bacteria, the virulent kind, alive and thriving. Something from the dead bacteria had transformed the living harmless bacteria into killers. Some substance, some molecule, had carried the instructions for virulence from one cell to another. Griffith called this the transforming principle, and he had no idea what it was. It took another 16 years before Oswald Avery, Colin MacLeod, and Macklin McCarty at the Rockefeller Institute, the very same institution where Rouse had worked with his chickens, finally identified the transforming principle as deoxyribonucleic acid, DNA. Their 1944 paper was a landmark, though like so many landmarks in this story, it was met with skepticism. Many scientists still believed that proteins, which are far more complex and varied than DNA, must be the carriers of genetic information. DNA seemed too simple, too monotonous, just 4 chemical bases repeating over and over. How could something so plain carry the instructions for building an entire organism? That question was answered in 1953 when James Watson and Francis Crick, working at the Cavendish Laboratory in Cambridge, built their famous model of the DNA double helix. And yes, They famously celebrated by walking into the Eagle pub and announcing that they had discovered the secret of life, which is a wonderful bit of scientific bravado. But the crucial insight of their model wasn't just the shape, it was the implication. The two strands of the helix are complementary. Each base on one strand pairs with a specific base on the other, adenine with thymine, cytosine with guanine, which means that each strand contains enough information to reconstruct the other, which means DNA can copy itself, which means you have a mechanism for heredity, a way for a cell to duplicate its genetic information and pass it to its daughter cells. And suddenly, the whole picture starts to come into focus. If genes are made of DNA and DNA can be copied, then errors in copying, mutations, could change the instructions. And if those changed instructions happen to affect genes that control cell growth, you could end up with a cell that grows when it shouldn't, a cell that divides when it should be resting, a cell that ignores the signals telling it to stop. In other words, cancer. Now, this connection wasn't made overnight. It took years of painstaking work in the 1950s and 1960s to crack the genetic code, to figure out how the sequence of bases in DNA gets translated into proteins, which are the molecular machines that actually do things inside cells. But once that framework was in place, researchers could finally start asking the right questions about cancer. Not just what causes it, in the broad sense of radiation or chemicals or viruses, but what specific changes in the DNA lead to uncontrolled growth? And this brings us back to that chicken virus. Remember Rouse's sarcoma virus? By the 1960s, scientists had learned a lot more about how viruses work. They knew that viruses are essentially packages of genetic material. Some use DNA, others use RNA, wrapped in a protein coat. A virus infects a cell by injecting its genetic material, which then hijacks the cell's machinery to make more copies of the virus. But tumor viruses, like the Rouse sarcoma virus, seem to do something extra. They didn't just replicate and kill the cell. They changed the cell's behavior. They transformed it. The infected cell started growing uncontrollably, piling up into a tumor. Something in the virus's genetic material was actively instructing the cell to become cancerous. In the late 1950s and into the 1960s, Howard Tiemann, a young researcher at the University of Wisconsin, was studying exactly this. Tiemann was working with the Rouse sarcoma virus, and he proposed something radical: that the virus, which carried its genetic information as RNA, was somehow converting that RNA into DNA and inserting it into the host cell's own genome. This was heretical. The central dogma of molecular biology, as Francis Crick had articulated it, said that information flows from DNA to RNA to protein. It was supposed to be a one-way street. RNA to DNA? That was going backward. That wasn't supposed to happen. Tiemann was ridiculed for years. People called his idea impossible. But in 1970, working independently, both Tiemann and David Baltimore discovered the enzyme that made it possible: reverse transcriptase. This enzyme does exactly what Tiemann had predicted. It reads an RNA template and builds a DNA copy. The Rouse sarcoma virus was using reverse transcriptase to write itself into the chicken cell's own DNA. It was literally inserting its genes into the host genome, becoming a permanent part of the cell's genetic instructions. And among those viral genes was one that could drive the cell to become cancerous. That gene was called SRC, short for sarcoma. And it was the first oncogene ever identified, the first gene shown to be capable of causing cancer. But here's where the story takes its most astonishing turn. And it involves that quiet researcher I mentioned earlier, the one who would discover something that changes everything we thought we knew about where cancer comes from. His name was Harold Varmus, and he was working with a colleague named J. Michael Bishop at the University of California, San Francisco. And in the mid-1970s, they asked a question so simple, it was almost naive. Where did the SRC gene come from? Was it purely a viral gene, something the virus had invented on its own? Or did it come from somewhere else? They used a molecular probe, essentially a piece of the viral SRC gene tagged so it could be detected, and they went looking for it in normal, healthy cells, not infected cells, not cancerous cells, normal cells, chicken cells that had never been anywhere near the Rous sarcoma virus. And they found it. The gene was there, sitting quietly in the DNA of every normal chicken cell. And not just chickens, they found closely related versions of the gene in turkeys, in ducks, in fish, in mammals, in humans, in us, you and me. The SRC gene wasn't some alien thing the virus had cooked up in its own little genome. It was ours. It had been ours all along. The virus had, at some point in its evolutionary history, picked up a copy of a normal cellular gene, carried it away, and in the process, altered it just enough to turn it into something dangerous. The cancer gene came from inside the house. Let that sink in for a moment. For decades, researchers had been chasing the origin of cancer in external agents, viruses, chemicals, radiation, and those things absolutely mattered. But what Bishop and Varmus showed was that the raw material for cancer, the genetic machinery capable of driving a cell toward uncontrolled growth, was already present in every single cell of your body. It was part of the normal blueprint, part of the plan. The virus didn't create the cancer gene. It stole one from us, corrupted it, and handed it back. This discovery, published formally in the late 1970s, earned Bishop and Varmus the Nobel Prize in Physiology or Medicine in 1989. And it fundamentally reshaped how we think about cancer at the deepest level, because it meant we needed a new vocabulary. The original normal version of the gene, the one sitting peacefully in your cells right now, doing useful work, that became known as a proto-oncogene. Think of it as a law-abiding citizen. It has a job. In the case of SRC, that job involves signaling pathways that tell cells when to grow and divide. It's essential. You need it. Without genes like SRC doing their thing during embryonic development, you'd never have formed properly in the first place. These proto-oncogenes are part of the fundamental toolkit of life. But when a proto-oncogene gets mutated, when something changes its DNA sequence, or it gets duplicated too many times, or a control switch near it gets rearranged, it can become an oncogene. The law-abiding citizen becomes a rogue. Instead of sending carefully regulated growth signals, it starts screaming, grow, 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 all the time. Or it sends the signal at the wrong moment, or it can't be turned off. And that's one of the key ignition events in cancer. Here's a good way to think about it. Imagine a car's accelerator pedal. In normal operation, you press it when you want to go and you release it when you want to stop. A proto-oncogene is like that accelerator. It's part of the system. It's supposed to be there, and it works beautifully when it's regulated properly. An oncogene is what happens when that accelerator gets stuck to the floor. The car just goes. It doesn't matter that you've taken your foot off the pedal. Something has jammed the mechanism, and now the engine is revving full speed with no input from the driver. That's what a mutated oncogene does to a cell. It locks the growth signal in the on position. And once researchers understood this framework, they started finding oncogenes everywhere. The floodgates opened. Throughout the late 1970s and into the 1980s, lab after lab identified new proto-oncogenes that could be converted into oncogenes by specific mutations. One of the most important was RAS. The RAS gene, actually a family of related genes, including HRAS, KRAS, and NRAS, was first identified through work on rat sarcoma viruses, which is where the name comes from. And when researchers started looking at human tumors, they found mutated RAS genes in a staggering proportion of cancers. We now know that roughly 1 in 4 of all human cancers carries a mutation in one of the RAS genes. Pancreatic cancer. About 90% of cases have a KRAS mutation. Colorectal cancer, lung cancer, RAS mutations show up again and again. The RAS protein is a molecular switch. In its normal state, it flips between an active form and an inactive form. When a growth signal arrives at the cell surface, say, a growth factor binds to a receptor on the outside of the cell membrane, that signal gets relayed inward through a chain of molecular events, and at one point in that chain, the RAS protein gets switched on. It stays on briefly, passes the signal along, and then flips itself back off. Beautiful, elegant, tightly controlled. But the most common mutations in RAS break the off switch. The protein gets stuck in its active conformation. It's like a light switch that you can flip up, but can never flip back down. The growth signal is now permanent, even when no growth factor is present at the cell surface. The cell thinks it's constantly being told to divide. Now, here's the thing that makes cancer so insidious, and this is a concept we'll keep coming back to. A single oncogene activation by itself usually isn't enough to cause full-blown cancer. If it were, we'd all be riddled with tumors by the time we were 20, because mutations happen all the time. Every time your cells divide, and your body is producing millions of new cells every second, the DNA replication machinery makes mistakes. Most get corrected by proofreading enzymes. Some slip through. Occasionally, one of those uncorrected mistakes lands in a proto-oncogene and turns it on. But the cell has backup systems. Layers and layers of backup systems. And this is where the story gets even more interesting, because we need to talk about the other side of the equation. The brakes. If oncogenes are stuck accelerators, then tumor suppressor genes are the brakes. And the discovery of tumor suppressors is its own remarkable chapter in this story, one that runs in parallel with the oncogene work but comes from a completely different direction. The key figure here is a man named Alfred Knudsen, a physician and geneticist who in 1971, right around the same time that Tumor and Baltimore were discovering reverse transcriptase, proposed something called the two-hit hypothesis. Knudsen was studying retinoblastoma, a rare cancer of the eye that primarily affects young children. What was peculiar about retinoblastoma was that it came in two forms. Some children developed tumors in just one eye, usually appearing a bit later in childhood. But other children developed tumors in both eyes, often very early, sometimes even before birth. And the bilateral cases tended to run in families. Knudsen looked at the statistical patterns, the age of onset, the number of tumors, the family histories, and he proposed a beautifully simple model. He said, what if this cancer requires two mutations, two hits to the same gene? In the hereditary form, the child is born already carrying one defective copy of the gene in every cell of their body. They inherited it from a parent. They only need one more mutation, one more hit in any single retinal cell, and that cell loses all function of this gene and becomes cancerous. That's why these children get tumors early in both eyes, often multiple tumors. The probability of that second hit occurring in at least one retinal cell is very high, but in the non-hereditary sporadic form, the child starts with 2 normal copies of the gene. Both copies have to be knocked out by random mutations in the same cell. The chances of that happening are much lower, which is why sporadic retinoblastoma typically appears later in just one eye as a single tumor. 2 independent random events have to hit the same gene in the same cell. It's not impossible, but it's much less likely. The gene in question was eventually identified in the mid-1980s and named RB1 for retinoblastoma 1. And it was the first tumor suppressor gene ever characterized. Unlike oncogenes, which cause cancer when they're turned on or made overactive, tumor suppressor genes cause cancer when they're turned off or lost. They're protective. They're the cell's defense system. The RB protein, the product of the RB1 gene, normally acts as a gatekeeper at a critical checkpoint in the cell division cycle. It essentially holds the cell back from dividing until conditions are right. When both copies of RB1 are functional, the cell has a reliable brake pedal. When both copies are lost, that brake is gone. And this 2-hit model turned out to be a general principle. It applies to tumor suppressors across the board. You have 2 copies of most genes, 1 from each parent. For a tumor suppressor, losing 1 copy usually isn't catastrophic because the remaining copy can still produce enough functional protein to do the job. You've lost 1 brake pad, but you've still got 1 left, and it works. But if the second copy goes too, through another mutation, or through a deletion of that chunk of chromosome, or through a process called loss of heterozygosity, then the brake is truly gone, and the cell has lost one of its critical safeguards against uncontrolled growth. Perhaps the most famous tumor suppressor of all is one you've almost certainly heard of, even if you've never studied biology. It's called p53, and it's sometimes referred to as the guardian of the genome. The gene that encodes p53 is called TP53, and it is the single most commonly mutated gene in human cancer. Mutations in TP53 show up in roughly half of all human cancers. Half. Think about that number for a second. Across all the incredible diversity of cancer types, lung, breast, colon, liver, brain, bone, blood, in about half of them, this one gene is broken. And when you understand what p53 does, you understand why losing it is so catastrophic. The p53 protein is a transcription factor, which means it controls the activity of other genes. It sits at the center of an enormous web of cellular responses. Under normal conditions, p53 is kept at very low levels in the cell. It's constantly being made and then immediately tagged for destruction. It's there, but it's being actively suppressed. It's like a fire alarm that's always ready, but silent. But when the cell detects trouble, DNA damage from radiation, chemical exposure, replication errors, or even abnormal signals from an activated oncogene, p53 gets stabilized, it accumulates, it becomes active, and then it does several remarkable things. First, it can halt the cell cycle. It stops the cell from dividing, buying time for repair. It activates DNA repair pathways, calling in the molecular machinery that can fix the damage. And if the damage is too severe to repair, if the cell's genome is so compromised that it's dangerous to let this cell continue existing, p53 can trigger apoptosis, programmed cell death. The cell essentially dismantles itself in an orderly fashion, sacrificing itself for the good of the organism. It's one of the most profound acts in all of biology, when you think about it. A single cell, detecting that it has become a potential threat, choosing to die so that the body can survive. So when p53 is lost, all of those protective responses vanish. DNA damage goes unrepaired. Cells with broken genomes keep dividing. Mutations accumulate faster and faster. The genome becomes unstable, and that instability feeds on itself, generating more mutations, more chromosomal chaos, more opportunities for additional oncogenes to be activated, and additional tumor suppressors to be lost. Losing p53 doesn't just remove one break, it removes the entire emergency response system. And this is why TP53 mutations are so common in cancer, and why cancers that have lost p53 tend to be more aggressive and harder to treat. There's a condition called Lee-Fraumeni syndrome, named after Frederick Lee and Joseph Fraumeni, who described it in 1969. People with this syndrome inherit one defective copy of TP53 from a parent. They're born with only one functional copy of the guardian of the genome. And the consequences are devastating. They have an extraordinarily high lifetime risk of developing cancer, often multiple different cancers, often starting in childhood or young adulthood. Sarcomas, breast cancer, brain tumors, leukemia, adrenal cortical carcinomas. It's Knudson's two-hit model, playing out in the most heartbreaking way possible. These families carry the weight of a single missing gene across generations. By the late 1980s and into the early 1990s, the picture was coming together with breathtaking clarity. Cancer wasn't one disease. It was a process, a multi-step process of accumulated genetic damage. You needed oncogenes switched on and tumor suppressors switched off, and usually you needed several of each. The cell had to lose its brakes and have its accelerator jammed simultaneously, and probably also lose its steering and its airbags. This was formalized in a way that would become one of the most influential frameworks in all of cancer biology, and the two scientists who pulled it all together into a single coherent vision were Douglas Hanahan and Robert Weinberg, who in the year 2000 published a paper that would become one of the most cited papers in the history of biology. It was called The Hallmarks of Cancer, and it appeared in the journal Cell in January 2000, and what Hanahan and Weinberg did was something that sounds simple, but was actually profoundly difficult. They looked at everything that had been learned about cancer over the previous 25 years, all the oncogenes, all the tumor suppressors, all the signaling pathways, all the mouse models, all the clinical observations, and they asked a deceptively straightforward question. What does a cell actually need to do in order to become cancerous? Not which specific genes are mutated, because those vary wildly from cancer to cancer. But what are the functional capabilities that every cancer must acquire, regardless of which particular mutations get it there? And they distilled the answer down to 6 essential capabilities. 6 hallmarks. Think of it this way. If you imagine a normal cell as a well-behaved citizen living in a complex society, following rules, responding to signals from its neighbors, knowing when to work and when to rest and when to die, then a cancer cell is essentially a cell that has broken free from every one of those social contracts. And Hanahan and Weinberg identified 6 specific contracts that need to be broken. The first hallmark is sustaining proliferative signaling. Normal cells don't just divide whenever they feel like it. They wait for external signals, growth factors, that tell them it's time to divide. It's like needing permission. A cancer cell figures out how to generate its own permission slips. Maybe it produces its own growth factors. Maybe it has mutated receptors on its surface that are permanently switched on, firing grow signals into the cell interior even when no growth factor is present. Remember the RAS oncogene we talked about? That's exactly what a mutated RAS protein does. It sits in the signaling chain between the receptor on the cell surface and the machinery in the nucleus that initiates cell division, and when RAS is mutated, it gets stuck in the on position. The cell thinks it's constantly receiving growth signals, even when no one is sending them. The cell becomes self-sufficient in its own proliferation. The second hallmark is evading growth suppressors. This is the flip side. Normal tissues don't just have go signals, they also have stop signals. Neighboring cells, the extracellular matrix, various molecular breaks all send messages that say, slow down, stop dividing, you're crowding. And this is where tumor suppressors like RB and P53 come in. A cancer cell has to find a way to ignore or disable these stop signals. It's not enough to have the accelerator jammed. You also have to cut the brake lines. And as we've seen, losing both copies of a tumor suppressor gene is one of the most common ways this happens. The third hallmark is resisting cell death. And this one is really important, so let's sit with it for a moment. Your body has a built-in self-destruct mechanism in every single cell. It's called apoptosis, programmed cell death, and it's not some catastrophic explosion. It's an elegant, orderly dismantling. The cell shrinks, its DNA is neatly chopped up, its contents are packaged into little membrane-bound bubbles, and neighboring cells or immune cells quietly clean up the debris. No inflammation, no mess. It's like a building being carefully deconstructed brick by brick rather than demolished with dynamite. And this system exists for a very good reason. When a cell detects that something has gone seriously wrong, its DNA is badly damaged, its growth signals are contradictory, it's in a place it shouldn't be, apoptosis is supposed to kick in and eliminate that cell before it can cause trouble. It's one of the body's most powerful anti-cancer defenses. A cell that's on the road to becoming cancerous should, in theory, trigger its own self-destruct. But cancer cells learn to disable apoptosis. They overexpress survival proteins. They disable the sensors that detect damage. They cut the wiring between the damaged sensors and the self-destruct machinery. And once a cell can resist apoptosis, it becomes enormously dangerous, because now it can accumulate more and more mutations without being eliminated. It just keeps going. The fourth hallmark is enabling replicative immortality. Here's something most people don't realize. Even if you took a normal human cell and gave it all the growth signals it wanted and removed all the breaks, it still wouldn't divide forever. Normal cells have a built-in counter, a molecular clock that limits how many times they can divide. And that clock is tied to structures called telomeres, which are repetitive stretches of DNA at the tips of your chromosomes. Every time a cell divides and copies its DNA, the telomeres get a little bit shorter. Think of them like the plastic aglets on the ends of shoelaces. They protect the chromosome ends from fraying. But they erode with each division, and after 50 or 60 divisions, the telomeres get critically short, and the cell enters a state called senescence. It stops dividing permanently. It's still alive, but it's retired. This is actually thought to be one of the body's anti-cancer mechanisms, a hard limit on how many times any cell lineage can replicate. But cancer cells get around this. Most cancer cells activate an enzyme called telomerase, which rebuilds the telomeres after each division. Telomerase is normally active in stem cells and germ cells, cells that genuinely need to divide indefinitely, but it's switched off in most adult cells. Cancer cells switch it back on. And with telomerase active, The replicative clock is reset every time, and the cell becomes, for all practical purposes, immortal. It can divide without limit. The fifth hallmark is inducing angiogenesis. And this one is beautifully practical, so let me paint the picture. Imagine a tiny cluster of cancer cells, maybe a few hundred, maybe a few thousand, sitting in some tissue. They're dividing, they're ignoring stop signals, they're resisting apoptosis. But they have a problem. They need oxygen and nutrients, and they need to get rid of waste products. And the only way to do that, in a body, is through blood vessels. Now, the nearest blood vessel might be close enough to supply a very small cluster of cells through simple diffusion, oxygen and glucose just seeping through the tissue. But once that cluster grows beyond about 1 millimeter or 2 in diameter, diffusion isn't enough. The cells in the center start starving, so the tumor needs its own blood supply. And here's what's remarkable. Cancer cells learn to send out chemical signals, particularly a molecule called vascular endothelial growth factor, or VEGF, that recruit nearby blood vessels to sprout new branches that grow toward and into the tumor. The tumor literally coerces the body into building it its own private circulatory system. This process, angiogenesis, the formation of new blood vessels, was a concept championed by a surgeon named Judah Folkman, who first proposed in the early 1970s that tumors were angiogenesis dependent and that blocking angiogenesis could be a way to treat cancer. He was ridiculed for years. People called him crazy. The idea that you could starve a tumor by cutting off its blood supply seemed too indirect, too strange. But Folkman was right, and his persistence eventually led to an entire class of anti-cancer drugs, anti-angiogenic therapies that are used in clinics today. Bevacizumab, sold as Avastin, is one of the most well-known. It's a monoclonal antibody that binds to VEGF and blocks it, essentially trying to cut the supply lines to the tumor. And the sixth hallmark is activating invasion and metastasis. This is the one that makes cancer truly lethal. A tumor that just sat in one place and grew would be dangerous, sure, but in many cases it could be surgically removed. What makes cancer so deadly is its ability to spread. Cancer cells acquire the ability to break free from the primary tumor, chew through the surrounding tissue, enter the bloodstream or lymphatic system, travel to distant organs, and establish new colonies there. Metastasis. And this is an incredibly complex process because each step requires the cancer cell to acquire new capabilities. It has to loosen its attachments to neighboring cells, which means down-regulating molecules like E-cadherin, which is basically the glue that holds epithelial cells together. It has to produce enzymes called matrix metalloproteinases that degrade the extracellular matrix, the structural scaffolding between cells. It has to become motile, learning to crawl and squeeze through tissue. It has to survive in the bloodstream, which is a hostile environment for a cell that's used to being anchored in tissue. And then it has to arrive at a distant organ, exit the bloodstream, and somehow establish itself in a completely foreign microenvironment. The fact that cancer cells can do all of this is honestly astonishing from a biological standpoint. Most cancer cells that enter the bloodstream die. The process is incredibly inefficient, but a tumor sheds millions of cells, and it only takes a few successful colonizers to establish a metastatic site. And once cancer has metastasized to multiple organs, it becomes extraordinarily difficult to treat. So those are the 6 original hallmarks: sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, activating invasion and metastasis. And the beauty of this framework is that it's agnostic about specific genes. Different cancers achieve these hallmarks through different mutations, different pathways, different mechanisms. Lung cancer and colon cancer and breast cancer might look completely different at the genetic level, but functionally, they've all had to solve the same 6 problems. That's a powerful insight. It means that despite the bewildering diversity of cancer at the molecular level, there's an underlying logic, a shared playbook. Now, Hanahan and Weinberg didn't stop there. In 2011, they published an updated version of the paper, Hallmarks of Cancer, the Next Generation, and they added more. They added 2 additional hallmarks: deregulating cellular energetics, which is about how cancer cells rewire their metabolism to fuel rapid growth, often relying on a form of glucose metabolism called the Warburg effect, named after Otto Warburg, who first noticed in the 1920s that cancer cells consume glucose at an astonishing rate even when oxygen is plentiful. And the other new hallmark was avoiding immune destruction, which acknowledges something that had become increasingly clear, that the immune system is constantly surveilling for and eliminating abnormal cells, and that successful cancers must find ways to evade or suppress that immune response. They also identified 2 enabling characteristics that aren't hallmarks themselves, but make acquiring the hallmarks possible: genome instability and mutation, which is the accelerated accumulation of genetic changes, and tumor-promoting inflammation, which is the way chronic inflammation can create a microenvironment that nurtures cancer development. And this idea of the tumor microenvironment, the neighborhood the tumor lives in, not just the tumor cells themselves, was becoming one of the most important shifts in how scientists thought about cancer. Because for a long time, the focus had been almost exclusively on the cancer cell itself. What mutations does it have? What pathways are activated? But researchers were starting to realize that a tumor isn't just a ball of identical malignant cells. It's a complex ecosystem. There are cancer cells, yes, but there are also fibroblasts that have been co-opted to support the tumor, immune cells that have been tricked into helping rather than attacking, blood vessels that have been recruited, signaling molecules flooding the local environment. The tumor reshapes everything around it, and everything around it influences the tumor in return. It's a conversation, a negotiation, a corruption of normal tissue architecture. And understanding that conversation was going to be critical for the next great frontier in cancer treatment, because some of the most revolutionary therapies of the 21st century wouldn't target the cancer cell at all. They'd target the immune cells that had been fooled into looking the other way. And the story of how that happened begins with a researcher who spent years studying a molecule that most people thought was completely uninteresting. A molecule on the surface of T cells called CTLA4, and his name was James Allison. And to understand why his work changed everything, you need to know a little bit about who he was and where he came from, because his path to one of the most important breakthroughs in cancer history was anything but straightforward. Jim Allison grew up in Alice, Texas, a small town in the southern part of the state. And from an early age, he was the kind of kid who wanted to take things apart and figure out how they worked. He lost his mother to lymphoma when he was 11 years old. That's a detail that matters. Not because it's some neat narrative arc where childhood tragedy leads directly to a cure. Life isn't that clean. But because it gave him a visceral, personal understanding of what cancer does to families. He knew what it looked like. He knew what it felt like. And later in his career, when the scientific establishment pushed back against his ideas, when funding was hard to get and pharmaceutical companies weren't interested, That personal knowledge gave him a stubbornness that turned out to be essential. Allison was a T-cell biologist. That was his world. He studied the immune system's most important soldiers, T-cells, those white blood cells that patrol your body looking for things that shouldn't be there. Viruses, bacteria, cells that have gone wrong. And by the early 1990s, immunologists had worked out the basic mechanics of how a T-cell gets activated. It's a two-signal system, and it's worth understanding because it's the foundation for everything that comes next. So imagine a T-cell encountering a suspicious cell. Let's say a cell that's been infected by a virus, or a cell that started to look cancerous. The suspicious cell presents little fragments of its internal proteins on its surface, displayed on molecules called MHC proteins, like holding up an ID badge. The T-cell has a receptor, the T-cell receptor, that recognizes that badge. That's signal 1. It's the T-cell saying, I see something unusual here. But signal 1 alone isn't enough. The immune system is careful. It doesn't want T-cells going off half-cocked, attacking healthy tissue. So there's a second signal required, a co-stimulatory signal. The most important one involves a molecule on the T-cell surface called CD28, which binds to molecules called B7 on the surface of the antigen-presenting cell. When the T-cell gets both signals, recognition plus co-stimulation, it activates. It starts dividing, it starts killing, it mounts an immune response. Now here's where it gets interesting. In the mid-1990s, researchers identified another molecule on T-cells called CTLA4. And CTLA4 looked a lot like CD28, structurally similar, bound to the same B7 molecules on antigen-presenting cells. But it did the opposite thing. Instead of giving the T-cell a green light, CTLA4 acted as a brake. When CTLA4 engaged with B7, it sent an inhibitory signal that told the T-cell to stand down, to stop, to not attack. And this makes sense from an evolutionary perspective, right? You need brakes on the immune system just as much as you need an accelerator. Without brakes, T-cells would attack everything. Your own tissues, harmless proteins, food in your gut. Autoimmune disease is what happens when those brakes fail. So CTLA4 is part of a checkpoint system, a quality control mechanism that keeps immune responses proportional and prevents them from spiraling out of control. Most immunologists at the time saw CTLA4 as exactly that, a safety mechanism, important for preventing autoimmunity, interesting in a basic science kind of way, but not particularly relevant to cancer therapy. And this is where Allison's thinking diverged from nearly everyone else's. Because Allison looked at CTLA4 and asked a different question. He didn't ask, how does this molecule prevent autoimmunity? He asked, what if cancer is exploiting this brake? What if the reason T-cells don't destroy tumors, even though they can recognize them, is that CTLA4 is slamming on the brakes before they can finish the job? And what if we could release that brake? Think about what that means. For decades, the approach to cancer immunotherapy had been to try to rev up the immune system, give it more fuel, more targets, more stimulation. Inject cytokines like interleukin-2 to crank up immune activity. Develop cancer vaccines to teach the immune system what to attack. And those approaches had mostly been disappointing. They worked in a small number of patients, sometimes dramatically, but for most people they didn't do much. Allison's insight was that maybe the problem wasn't that the immune system needed more gas. Maybe the problem was that the brakes were on, and instead of stepping harder on the accelerator, you needed to release the brake pedal. In 1996, Allison and his team published a landmark paper. They had developed an antibody that blocked CTLA-4, physically prevented it from engaging with B7, essentially cutting the brake line on T cells, and they tested it in mice with tumors. The results were striking. Mice that received the anti-CTLA-4 antibody rejected their tumors. Their immune systems, freed from the CTLA-4 checkpoint, attacked and destroyed the cancer. Some of those mice were completely cured. Now, curing cancer in mice is one of those things that happens all the time in research and almost never translates to humans. There's an old joke in oncology. We've cured cancer in mice 1,000 times. So healthy skepticism was warranted. But Allison believed this was different because he wasn't targeting the tumor. He wasn't relying on the tumor having a specific mutation or expressing a specific protein. He was targeting the immune system's own regulatory machinery. He was unleashing something that was already there, the body's natural ability to recognize and kill cancer, by removing the restraint that was holding it back. Getting the pharmaceutical industry to listen was another story entirely. Allison spent years trying to convince drug companies to develop a human version of his anti-CTLA-4 antibody. Most of them weren't interested. Immunotherapy had a bad reputation. Too many failed trials. Too many overpromised results. The smart money in the late 1990s and early 2000s was on targeted therapies. Drugs like imatinib, which we'll come back to, that went after specific molecular targets in cancer cells. The idea of treating cancer by manipulating the immune system felt old-fashioned to many people, almost fringe. But eventually, a small biotech company called Medarex took the gamble. They developed a fully human anti-CTLA-4 antibody called ipilimumab, and it entered clinical trials. The early results were confusing, honestly. The drug didn't behave like a traditional cancer therapy. With chemotherapy or targeted therapy, you expect to see tumors shrink relatively quickly if the drug is working. With ipilimumab, some patients' tumors initially seemed to grow before they started shrinking. Some patients developed new lesions that later disappeared. The immune system works on its own timeline, and the standard criteria for evaluating cancer drug responses weren't designed for this kind of therapy. But here's the thing that made people sit up and pay attention. In a phase 3 clinical trial for advanced melanoma, one of the deadliest forms of skin cancer, with a median survival at the time of about 6 to 9 months for metastatic disease, ipilimumab extended overall survival. The improvement was modest in absolute terms. Median survival went from about 6 months to about 10 months. That doesn't sound like much, right? But look at the survival curves. What was remarkable wasn't the median. It was the tail of the curve. About 20% of patients treated with ipilimumab were still alive years later. Some of them were alive 5 years later, 10 years later. In a disease where almost nobody survived long-term, a significant minority of patients seemed to be experiencing durable, lasting responses. Their cancers weren't just shrinking temporarily, they were staying gone. Ipilimumab was approved by the FDA in 2011 for the treatment of advanced melanoma. It was the first immune checkpoint inhibitor, and it validated an idea that James Allison had been championing for 15 years, that you could treat cancer by taking the brakes off the immune system. But the CTLA-4 story was just the beginning, because around the same time, another researcher on the other side of the Pacific was working on a different immune checkpoint molecule, and his discovery would prove to be even more transformative. His name was Tasuku Honjo, an immunologist at Kyoto University in Japan, and in 1992, his lab had identified a gene they called PD1, programmed death 1. The name came from the context in which they found it. They were studying cells undergoing apoptosis, programmed cell death, and PD-1 was expressed in those dying cells. But figuring out what PD-1 actually did took years of painstaking work. By the late 1990s, Hanjo's group had determined that PD-1, like CTLA-4, was an inhibitory receptor on T cells. It was another break, but it worked differently. While CTLA-4 mainly acts early in T cell activation, in the lymph nodes during the initial priming phase, PD-1 acts later, out in the tissues, at the site where T cells are trying to do their work. And PD-1's binding partner, a molecule called PDL1, turned out to be expressed on the surface of many cancer cells. Think about what that means. The cancer cells themselves were displaying the off-switch ligand on their surface. When a T cell arrived at the tumor, ready to kill, it would see PDL1 on the cancer cell. PD-1 on the T cell would engage with it, and the T cell would receive a signal to stand down. The cancer was essentially holding up a fake badge that said, "Don't shoot, I'm one of you." It was co-opting the immune system's own safety mechanism to protect itself. And this is where the tumor microenvironment story we talked about earlier comes full circle. Remember that idea of the tumor as an ecosystem, a corrupted neighborhood? PDL. One expression is one of the tools tumors use to corrupt that neighborhood. Some tumors upregulate PDL1 in direct response to immune attack. When T cells show up and start releasing inflammatory signals like interferon gamma, the tumor cells respond by cranking up PDL1 expression, essentially adapting in real time to shield themselves. It's a form of immune evasion that's almost elegant in its simplicity. Blocking PD-1 or blocking PDL1 turned out to produce clinical results that were even more impressive than blocking CTLA-4. The first anti-PD-1 antibodies to reach clinical trials, nivolumab, developed by Bristol Myers Squibb, and pembrolizumab, developed by Merck, showed response rates in melanoma, lung cancer, kidney cancer, and other tumor types that were unprecedented for immunotherapy. And the side effect profile was generally more manageable than ipilimumab, which could cause severe autoimmune reactions because CTLA-4 blockade is a broader, more systemic removal of immune restraint. PD-1 blockade was more targeted, more focused on the tumor microenvironment where PDL1 was being expressed. Pembrolizumab received accelerated FDA approval in 2014 for advanced melanoma, and nivolumab followed shortly after. Within a few years, anti-PD-1 and anti-PDL1 drugs had been approved for dozens of cancer types: lung cancer, bladder cancer, head and neck cancer, Hodgkin lymphoma, liver cancer. The list kept growing. In 2018, James Allison and Tasuku Honjo shared the Nobel Prize in Physiology or Medicine for their discovery of cancer therapy by inhibition of negative immune regulation. It was one of those Nobel Prizes where most people in the field said, "Finally, that was overdue." But here's what's important to understand about checkpoint inhibitors, because the story isn't all triumph. These drugs are genuinely revolutionary. They have saved lives that would have been lost. There are people walking around today, years after a diagnosis of metastatic melanoma or advanced lung cancer, who are alive because of these drugs. But they don't work for everyone. In most cancer types, checkpoint inhibitors help somewhere between 20 and 40% of patients. For the rest, the drugs don't produce a meaningful response. And figuring out why some patients respond dramatically and others don't respond at all, became one of the most urgent questions in oncology. Part of the answer has to do with something called tumor mutational burden. Cancers that have a lot of mutations tend to respond better to checkpoint inhibitors, and the logic makes sense when you think about it. More mutations mean more abnormal proteins, which means more neoantigens, novel fragments that the immune system can recognize as foreign. A highly mutated tumor is basically waving more red flags, giving T cells more targets to lock onto once the brakes are released. Melanoma and lung cancer, two of the cancer types that respond best to checkpoint inhibitors, tend to have very high mutational burdens. Melanoma because of UV radiation damage, lung cancer because of the mutagenic effects of tobacco smoke. Cancers with lower mutational burdens, like many pancreatic cancers, tend to respond poorly. Their surfaces are too quiet, immunologically speaking. The T cells don't have enough to grab onto. Another piece of the puzzle involves the tumor microenvironment itself. Some tumors are described as hot. They're already infiltrated with T cells that are present but being held in check by PD-L1 and other immunosuppressive signals. For these tumors, releasing the checkpoint can unleash an army that's already in position. Other tumors are cold. The T cells never made it into the tumor in the first place. The tumor has built walls, figuratively speaking, that keep immune cells out. For cold tumors, simply releasing the brakes isn't enough because there's nobody in the driver's seat. And converting cold tumors into hot ones, finding ways to get T cells into the tumor microenvironment so that checkpoint inhibitors can then do their work, is one of the major areas of active research right now. And then there's the question of combination therapy, because oncologists quickly realized that combining checkpoint inhibitors with each other or with other treatments could sometimes produce better results than any single agent alone. Combining ipilimumab with nivolumab, blocking both CTLA-4 and PD-1 simultaneously, releasing two different brakes at once, showed impressive response rates in melanoma and kidney cancer, though at the cost of significantly more side effects. Combining checkpoint inhibitors with chemotherapy, with radiation, with targeted therapies, all of these approaches are being tested in hundreds of clinical trials around the world right now. But immunotherapy isn't just about checkpoint inhibitors. Around the same time that ipilimumab and nivolumab were transforming oncology, another approach was emerging that took immune manipulation to an entirely different level, one that involved removing a patient's own T cells from their body, genetically engineering those cells in a laboratory to recognize cancer, and then infusing them back into the patient as a living drug. It sounds like science fiction, and honestly, the story of how it went from a radical idea to an FDA-approved therapy involves one of the most dramatic clinical trials in the history of medicine. And that story really begins with a man named Carl June. Carl June is a physician-scientist at the University of Pennsylvania, and his path to becoming one of the architects of CAR T-cell therapy is one of those winding, deeply personal journeys that reminds you how much of science is shaped by the lives of the people doing it. June trained as an immunologist in the Navy, and early in his career, he worked on HIV research, trying to understand how to boost T-cell function in patients whose immune systems were being destroyed by the virus. That work gave him deep expertise in T-cell biology, how to grow T-cells outside the body, how to activate them, how to keep them alive and functioning. But his interest in cancer became intensely personal when his wife was diagnosed with ovarian cancer in 1996. She went through standard treatments, and June watched the limitations of conventional oncology from the other side of the doctor-patient divide. She passed away in 2001, and by that point, June had already been working on an idea that would eventually change everything. The concept behind CAR T is beautifully logical once you understand it, so let's walk through it slowly. Your T cells are extraordinary cancer fighters, as we've been talking about. But the problem is recognition. A T cell can only attack what it can see, and it sees through its T cell receptor. That molecular sensor on its surface that recognizes specific fragments of protein displayed on other cells. The trouble with many cancers is that the T cell receptors in your body might not recognize the tumor well enough, or the tumor has found ways to hide, or the T cells that could recognize it have been suppressed or exhausted. So what if you could skip all of that complexity? What if you could take a patient's T cells out of their body, give those cells an entirely new receptor, one that you've designed in a lab to recognize a specific protein on the surface of cancer cells, and then put those souped-up cells back into the patient? That's exactly what a CAR T cell is. CAR stands for chimeric antigen receptor. Chimeric because it's an artificial fusion, a receptor that doesn't exist in nature, stitched together from parts of different proteins. The outside portion of the CAR is typically derived from an antibody fragment, chosen because it binds tightly to a specific molecule on the cancer cell's surface. The inside portion connects to the T cell's internal signaling machinery so that when the outside part grabs its target, the T cell activates. It wakes up, it multiplies, it kills. You're essentially bolting a new targeting system onto an existing weapon. Now, the idea of engineering T cells to attack cancer wasn't entirely new. An Israeli researcher named Zelig Eshar had developed the first chimeric receptor concept back in the late 1980s. But those early designs didn't work well in patients because the engineered T cells would activate briefly and then fizzle out. They didn't persist in the body. They didn't multiply. They were a spark without a fire. The breakthrough that made CAR T actually work came from adding what are called costimulatory domains to the intracellular part of the receptor. Think of it this way. A T cell needs more than just the initial go signal to mount a sustained attack. It needs a second signal, a confirmation that tells it to keep going, to proliferate, to survive. In normal immune responses, this second signal comes from other molecules on the surface of antigen-presenting cells. The clever trick was to build that second signal directly into the CAR itself, so the engineered T cell gets both signals, activation and persistence, the moment it contacts its target. Michel Sadelain at Memorial Sloan Kettering and Carl June's group at Penn were among the key teams that developed these second-generation CARs, and that's what made the whole approach viable. The target they chose for the first major clinical trials was a protein called CD19, and this is important because it illustrates a key challenge in CAR T therapy, finding the right target. CD19 is a protein found on the surface of B cells, which are a type of white blood cell. In B cell cancers, certain leukemias and lymphomas, the malignant cells are covered in CD19. So if you engineer T cells to attack anything displaying CD19, they'll go after the cancer. The catch is that normal, healthy B cells also display CD19, so the CAR T cells will destroy those too. But here's the thing. You can live without B cells. It's not ideal, and patients need ongoing treatment to replace the antibodies that B cells would normally produce, but it's survivable. That trade-off, killing normal B cells as collateral damage in order to eliminate the cancer, was deemed acceptable. And that's a really important concept in CAR T therapy. You need a target that's present on the cancer, but where destroying normal cells that share that target won't kill the patient. For solid tumors, finding such targets has been enormously difficult, and that's one of the big reasons CAR T has been most successful in blood cancers so far. But let me tell you about the trial that put CAR T on the map. because it's one of those stories that gives you chills. In 2012, Carl June's team at Penn treated a 6-year-old girl named Emily Whitehead. Emily had acute lymphoblastic leukemia, ALL, the most common childhood cancer. She'd been through 2 rounds of chemotherapy, and both times the cancer came back. She'd run out of standard options. Her family enrolled her in the CAR-T trial, and her T cells were extracted, engineered to target CD19, and infused back into her body. What happened next was terrifying. Within days, Emily became critically ill. Her temperature spiked to over 106 degrees. Her blood pressure plummeted. Her organs started to fail. She was put on a ventilator in the pediatric ICU, and her medical team genuinely thought she might not survive the night. What was happening was something called a cytokine storm. The engineered T cells were working. They were attacking the cancer with incredible ferocity. But in the process, they were releasing massive quantities of inflammatory signaling molecules called cytokines. It was like setting off a bomb inside her immune system. The inflammation was so severe, it was killing her. And here's where a piece of luck, or maybe you'd call it prepared minds meeting opportunity, changed everything. June's team ran blood tests and found that one particular cytokine, interleukin-6, was elevated to an astronomical degree. And June's daughter happened to have juvenile rheumatoid arthritis, so he knew that there was an FDA-approved drug called tocilizumab that blocks interleukin-6 signaling. It had been developed for autoimmune conditions, not cancer. But in a desperate, last-ditch decision, the team administered tocilizumab to Emily. Within hours, her fever broke. Her blood pressure stabilized. She woke up. It was her 7th birthday. And when they tested her bone marrow afterward, the leukemia was gone. Completely gone. Her CAR-T cells had eliminated every detectable cancer cell in her body. Emily Whitehead became the first pediatric patient to be treated with CAR-T cell therapy, and as of now, more than a decade later, she remains cancer-free. She's a teenager. She's healthy. And the cytokine release syndrome that nearly killed her is now a recognized and manageable side effect of CAR-T therapy, in large part because of what her medical team learned that night. The FDA approved the first CAR-T therapy, tisagenlecleucel, marketed as Kymriah, in August 2017 for pediatric and young adult patients with relapsed B-cell ALL. 2 months later, a second CAR-T product, axicabtagene ciloleucel, was approved for certain types of aggressive B-cell lymphoma. Since then, several more CAR-T products have been approved for various blood cancers, including multiple myeloma. But let's be honest about the limitations, because this is not a miracle cure for all cancers, at least not yet. CAR-T therapy is extraordinarily expensive. We're talking several $100,000 per treatment, and that's before the cost of hospitalization and managing side effects. The manufacturing process is complex and individualized. You're making a custom therapy for each patient from their own cells, which takes weeks. Cytokine release syndrome, while now manageable, can still be dangerous. There's another serious side effect called neurotoxicity, confusion, seizures, difficulty speaking, that can occur and whose mechanism isn't fully understood. And as I mentioned, CAR-T has been most effective against blood cancers where you can identify a clean surface target like CD19. Solid tumors, the breast cancers, lung cancers, colon cancers that kill the most people, have been much harder to crack with this approach. Solid tumors don't always have a single uniform surface marker to target. They create hostile microenvironments that suppress T cell function even after the cells arrive. They build physical barriers of dense tissue that T cells struggle to penetrate. so the field is working on all of these problems simultaneously. Researchers are developing CAR T cells that can target multiple antigens at once, so the cancer can't escape by simply losing one surface protein. They're engineering T cells that secrete their own cytokines to counteract the immunosuppressive tumor microenvironment. They're exploring ways to make off-the-shelf CAR T cells from donor T cells rather than requiring each patient's own cells, which could dramatically reduce cost and manufacturing time. Some groups are even moving beyond T cells entirely, engineering natural killer cells or other immune cells with chimeric receptors. And this brings us to something worth stepping back and appreciating for a moment. Because when you look at where cancer treatment stands right now compared to where it was even 30 years ago, the transformation is staggering. We've gone from a world where the only weapons were surgery, radiation, and cytotoxic chemotherapy, essentially cutting, burning, and poisoning, to a world where we can decode the specific genetic mutations driving an individual patient's tumor, design drugs that target those exact molecular abnormalities, unleash the patient's own immune system by releasing molecular breaks, and even genetically engineer immune cells to hunt cancer with a precision that would have seemed like fantasy a generation ago. And yet, cancer still kills nearly 10 million people worldwide every year. So the question becomes, with all of these extraordinary tools, why haven't we won yet? And the answer to that question takes us into what might be the most important concept in modern cancer biology, something that explains why cancer is not one disease but hundreds, why treatments that work brilliantly in one patient fail completely in the next, and why the future of oncology is being built around a word that you've probably heard but might not fully appreciate the depth of. And that word is heterogeneity. Tumor heterogeneity. It sounds like a clinical term, something you'd find buried in a journal article, but it's actually the single most important reason cancer remains so difficult to defeat. And once you understand it, everything else about modern oncology, why treatments fail, why cancers come back, why your oncologist might sequence your tumor's DNA before choosing a drug, all of it clicks into place. So let's talk about what heterogeneity really means, because it operates on levels that are genuinely surprising. The first level is the one most people intuitively grasp. Different types of cancer are different diseases. Breast cancer is not the same disease as pancreatic cancer. They arise in different tissues, they're driven by different mutations, they grow at different rates, they respond to different treatments. That much seems obvious. But the depth of the differences is more profound than you might expect. Even within a single category, say breast cancer, there are molecularly distinct subtypes that behave so differently they might as well be separate diseases. A hormone receptor-positive breast cancer that's driven by estrogen signaling is a fundamentally different biological entity from a HER2 positive breast cancer that's driven by overexpression of a growth factor receptor, which is in turn a completely different beast from triple-negative breast cancer, which lacks all three of the standard receptor targets and tends to be more aggressive and harder to treat. These aren't subtle shadings of the same disease. They require different drugs, carry different prognoses, and arise through different molecular pathways. And this is just breast cancer. One organ, one general category, and already we're looking at what amounts to several distinct diseases wearing the same name. Now multiply that across every tissue in the body. Lung cancer has its own constellation of molecular subtypes. Tumors driven by EGFR mutations, ALK rearrangements, KRAS mutations, ROS1 fusions, and on and on. Colorectal cancer has its subtypes, melanoma has its subtypes, Each one responds to different targeted therapies or doesn't respond at all. This is why the old model of treating cancer primarily by where it started in the body. You have lung cancer. Here's the lung cancer protocol. Has been gradually giving way to something more nuanced, more molecular. Increasingly, oncologists care less about where the tumor lives and more about what's driving it at the genetic level. There's actually a fascinating concept that's emerged from this realization, sometimes called tumor-agnostic treatment. In 2017, the FDA approved pembrolizumab. That's the anti-PD-1 checkpoint inhibitor we talked about earlier, for any solid tumor, regardless of where it originated, as long as it had a specific molecular feature. High microsatellite instability, or deficient mismatch repair. Think about what that means. For the first time, a cancer drug was approved not for breast cancer, or colon cancer, or stomach cancer, but for a molecular signature that could appear in any of them. It didn't matter where the tumor was. What mattered was how its DNA behaved. That was a genuine paradigm shift, and it happened because of our deepening understanding of heterogeneity between cancer types. But here's where it gets really interesting, and honestly, this is the part that keeps researchers up at night. Heterogeneity doesn't just exist between different patients. It exists within a single patient's tumor. A single mass growing inside someone's lung or liver isn't a uniform ball of identical cancer cells. It's more like a city, a complex, diverse ecosystem where different neighborhoods contain cells with different mutations, different behaviors, different vulnerabilities. One region of the tumor might carry a mutation in KRAS. Another region might have amplified MYC. A third might have lost P53 function, while the others haven't. These subpopulations coexist, compete with each other for resources, and, here's the critical part, respond differently to treatment. Imagine you're an oncologist, and you give a patient a targeted drug that's beautifully effective against cells with a specific EGFR mutation. And it works. The tumor shrinks dramatically. The scans look great. Everyone's hopeful. But hidden within that tumor was a tiny subpopulation of cells, maybe 1% or less, that didn't carry that particular EGFR mutation, or that carried an additional mutation conferring resistance. Those cells weren't affected by the drug at all. And now, with all their competitors killed off, they have the run of the place. They multiply. They expand. And 6 months or a year later, the cancer is back. Except now it's composed almost entirely of resistant cells, and the drug that worked so well before is useless. This is the fundamental tragedy of cancer treatment, and it plays out over and over again. It's not that our drugs don't work. Many of them work brilliantly, for a while. The problem is evolution. And I mean that in the most literal Darwinian sense. A tumor is a population of cells under selective pressure, and when you apply a treatment, you're applying a selection force. You kill the sensitive cells and leave behind the resistant ones. It's natural selection happening in real time, inside a patient's body, on a timescale of weeks or months rather than millennia. Cancer doesn't outsmart our drugs through some kind of malicious intelligence. It out-evolves them through the blind mechanical logic of variation and selection. This evolutionary perspective on cancer has become one of the most important frameworks in modern oncology, and it owes a lot to a researcher named Robert Gatenby at the Moffitt Cancer Center in Florida. Gatenby, who trained in both radiology and mathematical biology, started arguing in the early 2000s that oncologists were making a fundamental strategic error. The standard approach was, and in many cases still is, to hit the cancer as hard as possible with the maximum tolerated dose of a drug, trying to kill every last cancer cell. The goal was total eradication. Gatenby pointed out that this is actually the worst possible strategy if your enemy can evolve resistance, because maximum dose treatment applies maximum selective pressure. You wipe out all the drug-sensitive cells as fast as possible, eliminating the very population that was competing with and suppressing the resistant cells. You're essentially clearing the field for the resistant clones to take over. His alternative proposal was something he called adaptive therapy, and it's counterintuitive enough that it initially met a lot of skepticism. The idea is this: instead of trying to kill every cancer cell, you use treatment to control the tumor population, keeping it stable rather than trying to eliminate it. You treat until the tumor shrinks to a certain level, then you back off. You let the sensitive cells regrow and reestablish themselves, because those sensitive cells compete with the resistant cells for space and nutrients. Then you treat again. You're essentially managing the tumor like an ecosystem, using the drug-sensitive cells as allies to keep the resistant population in check. You're not curing the cancer. You're turning it into something more like a chronic, manageable condition. Now, this is still a relatively new and somewhat controversial approach, and it's not appropriate for every situation. There are cancers where aggressive treatment genuinely can achieve a cure, and you absolutely want to go for it. But for metastatic cancers where cure is unlikely, adaptive therapy offers a different philosophy, one rooted in ecological and evolutionary thinking, rather than the traditional military metaphor of total war. Early clinical trials, particularly in metastatic prostate cancer, have shown promising results, with patients on adaptive therapy maintaining stable disease for significantly longer than patients on continuous maximum dose treatment, while also experiencing fewer side effects and better quality of life. It's still being studied, but the underlying logic is compelling precisely because it takes heterogeneity and evolution seriously. And this evolutionary lens connects to something else that's reshaping how we think about cancer: the concept of liquid biopsy. Traditionally, if you wanted to know what mutations were driving a patient's tumor, you needed a tissue biopsy. A surgeon or interventional radiologist would stick a needle into the tumor, pull out a sample, and send it to the lab for genetic sequencing. The problem is, a needle biopsy only samples one small region of what might be a highly heterogeneous tumor. You might miss the resistant subclone entirely, and you can't easily repeat the procedure every few weeks to track how the tumor is evolving in response to treatment. But here's what's clever. Cancer cells, as they grow and die, shed tiny fragments of their DNA into the bloodstream. This is called circulating tumor DNA, or ctDNA. And with modern sequencing technology, we can now detect and analyze those fragments from a simple blood draw. A tube of blood. That's it. And because the circulating DNA comes from cells throughout the tumor, and from metastases in different parts of the body, it actually gives you a more comprehensive picture of the tumor's genetic diversity than a single needle biopsy ever could. You can track mutations appearing and disappearing over time. You can detect resistance mutations emerging weeks or months before they'd show up on a scan. You can monitor whether a treatment is working at the molecular level in something close to real time. The first liquid biopsy tests started receiving FDA approval around 2016 and 2017, and the technology has been improving rapidly ever since. Companies like Guardant Health and Foundation Medicine have developed panels that can screen for hundreds of cancer-related mutations from a single blood sample. And the implications go beyond just monitoring treatment response. There's been enormous interest in using liquid biopsy for early detection, catching cancer before it's even symptomatic. when it's still at a stage where treatment is most likely to succeed. A company called Grail, which spun out of the genomics giant Illumina, developed a test called Gallery that screens for over 50 types of cancer from a single blood draw by detecting methylation patterns in circulating DNA. It launched commercially in 2021, and while it's not yet sensitive enough to replace standard screening methods for specific cancers, the concept is extraordinary. A single blood test that could flag dozens of cancer types at their earliest, most treatable stages. And all of this, the targeted therapies, the immunotherapies, the liquid biopsies, the evolutionary treatment strategies, all of it feeds into what has become the defining ambition of 21st century oncology, the thing that ties every thread we've been following together into a single coherent vision. It's the idea that every patient's cancer is unique, that treatment should be tailored to the specific molecular profile of that individual's disease, and that we should be monitoring and adapting in real time as the cancer evolves. The word for this, of course, is precision medicine, and while it's become something of a buzzword, the substance behind it is real, and it's transforming outcomes for patients in ways that would have been unimaginable even 20 years ago. Think about what oncology looked like at the turn of the millennium. If you were diagnosed with, say, non-small cell lung cancer in the year 2000, your oncologist had a handful of chemotherapy regimens to choose from, and the choice was based mostly on the type of tissue the cancer came from and how far it had spread. That was about it. The 5-year survival rate for advanced lung cancer was in the low single digits. It was grim. And the treatment itself was brutal. Platinum-based chemotherapy that attacked every rapidly dividing cell in your body, cancer or not, leaving patients exhausted, immunocompromised, nauseated, and often wondering whether the cure was worse than the disease. Now fast forward to today. That same patient walks into an oncologist's office, and the first thing that happens is molecular profiling. A biopsy sample, or increasingly, a blood draw, gets sent to a lab where it's screened for dozens of specific genetic alterations. Does this tumor carry an EGFR mutation, an ALK rearrangement, AROS1 fusion, a BRAF-V600E mutation, AMET exon 14 skipping alteration? Is there high PDL1 expression? What's the tumor mutational burden? Each of those results opens a different treatment pathway, a different drug or combination of drugs designed to exploit the specific vulnerabilities of that particular cancer. And the outcomes have changed dramatically. Patients with ALK rearranged lung cancer, which accounts for maybe 3 to 5% of non-small cell cases, now have access to drugs like alectinib and lorlatinib, ALK inhibitors that can keep the disease controlled for years, sometimes many years, with side effects that are manageable enough that people continue working, traveling, living their lives in ways that would have been unthinkable in the era of one-size-fits-all chemotherapy. That's not a cure, not yet, not for most of them. But it's a transformation in what it means to live with cancer. And lung cancer is just one example. The same molecular revolution has reshaped treatment across dozens of cancer types. In melanoma, the combination of BRAF and MEK inhibitors, drugs like dabrafenib and trametinib, can produce rapid, dramatic responses in patients whose tumors carry BRAF-V600 mutations. In breast cancer, the development of CDK4/6 inhibitors like palbociclib, first approved in 2015, extended progression-free survival for patients with hormone receptor-positive HER2-negative advanced breast cancer by nearly a year compared to hormonal therapy alone. In chronic myeloid leukemia, which we touched on earlier with imatinib, the story is even more dramatic. What was once a death sentence has become a chronic condition managed with a daily pill, and some patients who achieve deep molecular responses can even stop treatment and remain in remission. Here's what's really striking about all of this, though. It's not just that we have better drugs. It's that the entire logic of how we think about cancer treatment has shifted. The old model was categorical. You have breast cancer, here's the breast cancer protocol. The new model is molecular. You have a cancer with these specific mutations. Here's the treatment that targets those mutations, regardless of where in the body the cancer originated. Remember that landmark 2017 FDA approval we talked about, where pembrolizumab was approved for any solid tumor that was microsatellite instability high? That was the first time the FDA approved a cancer drug based purely on a biomarker rather than a tumor's anatomical location. And it wasn't the last. In 2018, larotrectinib became the second tumor-agnostic approval, targeting cancers with NTRK gene fusions, rare fusions that can show up in everything from salivary gland tumors to infantile fibrosarcoma to thyroid cancer. The response rates were remarkable, around 75% across tumor types, and the principle it established was profound. The molecular identity of a cancer can matter more than the organ it started in. Now, I want to be honest with you here, because precision medicine is genuinely exciting, but it's also genuinely complicated, and the reality on the ground is messier than the narrative sometimes suggests. Not every cancer has a targetable mutation. In fact, for many common cancers, the majority of patients don't have a clear molecular target that matches an available drug. Even when they do, resistance almost always develops eventually. The cancer evolves, finds a workaround, activates a bypass pathway, and the drug that was working stops working. This is that evolutionary pressure we talked about, the Darwinian logic of cancer playing out in real time inside a patient's body. And then there's the question of access. These targeted therapies and immunotherapies are expensive. We're talking tens of thousands, sometimes hundreds of thousands of dollars per year. Molecular profiling itself isn't free, and the infrastructure required to do this well, the sequencing technology, the bioinformatics expertise, the tumor boards where specialists sit down and interpret complex genomic reports together, that infrastructure isn't evenly distributed. A patient at a major academic cancer center in Boston or Houston has access to a very different level of precision oncology than a patient in a rural community hospital. That gap is real, and closing it is one of the major challenges of the next decade. But even with all those caveats, the trajectory is unmistakable. We are getting better at this, and the pace of improvement is accelerating in ways that genuinely surprise even the people working in the field. Let me give you a sense of what the cutting edge looks like right now, because some of it is frankly astonishing. One of the most exciting frontiers is the application of artificial intelligence to cancer diagnosis and treatment planning. Machine learning algorithms are being trained on enormous datasets, thousands of tumor genomic profiles, millions of pathology images, vast repositories of clinical outcomes. And they're starting to find patterns that human eyes and human minds simply can't detect at the same scale. In pathology, AI systems can now analyze digitized tissue slides and identify features that predict how a tumor will behave, sometimes picking up on subtle structural patterns in the tissue architecture that even experienced pathologists might miss. Google's DeepMind and other groups have developed models that can classify tumors, predict genetic mutations from histology images alone, meaning you might be able to infer something about a tumor's molecular profile just from looking at how the cells are arranged under a microscope before you even run a genetic test. It's early days, And these tools are being developed as aids to human pathologists rather than replacements, but the potential to speed up diagnosis and make high-quality interpretation available in resource-limited settings is enormous. And then there's the question that has haunted oncology from the very beginning, the question that keeps researchers up at night and drives patients and families to seek out every possible option. Can we actually cure cancer? Not manage it, not control it for a while, not extend survival by months, but cure it, eliminate it completely, make it go away and never come back? The honest answer is that we already can, for some cancers, in some patients. We've been curing certain cancers for decades. Many childhood leukemias, testicular cancer, early-stage Hodgkin lymphoma, some early-stage solid tumors caught before they spread. Surgery alone cures a significant fraction of early-stage cancers. The 5-year survival rate for localized breast cancer is over 99%. For localized prostate cancer, it's nearly the same. The problem has always been advanced cancer, metastatic cancer, cancer that has spread beyond its original site and seeded itself in distant organs. That's where the survival curves drop, where the treatments become more about buying time than achieving cure. But even here, things are shifting. Some patients with metastatic melanoma treated with checkpoint inhibitors are now more than 10 years out from treatment with no evidence of disease. Some of the children treated with CAR T therapy, like Emily Whitehead, are more than a decade past their treatment with no sign of leukemia returning. These are still the exceptions rather than the rule. But the fact that they exist at all, that some patients with cancers that were universally fatal 20 years ago are now living long, healthy, apparently cancer-free lives, that tells you something important about where the field is heading. And here's what I think is the most quietly revolutionary idea in modern cancer research, the one that doesn't get as many headlines but might matter the most in the long run. It's the idea of interception, catching cancer not just early, but before it's really cancer at all. We know now that the journey from a normal cell to a malignant tumor takes years, often decades. There are intermediate stages, precancerous lesions, clonal expansions of cells with 1 or 2 mutations, but not enough to become fully malignant. What if we could identify people in those intermediate stages and intervene? Not with chemotherapy, but with targeted prevention, drugs or vaccines or lifestyle interventions that reduce the probability of those precancerous cells ever completing the journey to full malignancy. This is already happening in some contexts. The HPV vaccine, which prevents infection with the strains of human papillomavirus that cause cervical cancer, is essentially a cancer prevention tool, and it's extraordinarily effective. Colonoscopy screening that finds and removes precancerous polyps prevents colorectal cancer from ever developing. The aspirin studies suggesting that regular low-dose aspirin might reduce the risk of certain cancers are another example, though the data there is more nuanced. The vision is to extend this principle much further, to use molecular profiling, liquid biopsy, and AI-driven risk models to identify individuals at the earliest possible stages of cancer development and intervene before the disease ever becomes clinically apparent. It's a beautiful idea, and it's one that connects all the way back to where we started this whole conversation, all those hours ago. Because when you understand cancer as a process, not a single event, but a slow, multi-step accumulation of genetic and epigenetic changes, shaped by environment and chance and the relentless logic of cellular evolution, then it makes sense that the best place to fight it might not be at the end of that process, when the tumor is established and diverse and adapted, but at the beginning, when the first few cells start to go wrong and the body's defenses might still be enough to hold the line with just a little help. And that, really, is the story of cancer. It's a story about cells and genes and the molecular machinery that keeps life running. It's a story about brilliant, stubborn, sometimes lucky people who spent their careers trying to understand something that seemed impossibly complex, and who, piece by piece, built a picture that now lets us fight back in ways that were pure science fiction within living memory. It's a story that isn't finished, not remotely, but one where the trajectory is unmistakably toward understanding, toward precision, toward hope, grounded in real science rather than wishful thinking. So as you lie there, maybe drifting, maybe already half asleep, let that settle over you. Not as a burden, not as something heavy, but as something quietly remarkable. That the same biological machinery that makes you alive, the DNA replicating, the cells dividing, the immune system patrolling, is the same machinery that researchers around the world are learning to read and repair and redirect. Every breath you take is millions of cells doing their jobs, and the vast, vast majority of the time, they get it right. The systems work. The proofreading catches the errors. The immune cells find the stragglers. And on the rare occasions when something slips through, we now understand, better than at any point in human history, what went wrong and how to respond. That understanding is still growing, still deepening, every single day, in labs and clinics and hospitals on every continent. And that's a good thing to fall asleep